the chairs of this session for allowing me to present the work performed in the nanomedicine laboratory at the Luigi Sacco University Hospital in Milan, Italy, where so far we have designed and characterized multifunctional nanoconstructs bearing antibody-derived ligand for breast cancer detection. Breast cancer, as you all know, is a target of primary interest from both a diagnostic and a therapeutic point of view, since it is the 5th most common cause of cancer death worldwide and the most frequent and persistent malignant cancer in females. At present, a crucial issue in breast cancer diagnostics is the identification of malignant tumors at early stages, but also track possible migration of tumor cells and metastatic infiltrations, principally at the axillary nodes, as well as monitor tumor regression and tumor response to surgical or pharmacological treatments. During last decades, magnetic nanoparticles have been proposed and used in clinics as innovative agents for non-invasive, easy, and reliable detection of cancer sites, and in particular, iron oxide-based nanoparticles are able to enhance the contrast by magnetic resonance imaging thanks to their superparamagnetic properties. Nanoparticles accumulation in the tumor mass can be driven in a passive manner by the wide fenestrations present at the endothelial vessels in close proximity to the tumor mass, following a phenomenon called enhanced permeability and retention effect. However, different nanomaterials also offer the possibility of a surface functionalization with different targeting agents, which could trigger a selective recognition of cancer cells and not of normal cells. And in this presentation, I would like to focus on this active targeting mechanism triggered by OMICS ligands immobilized onto the surface of magnetic nanoparticles. And in order to do that, I will focus on the human epidermal growth factor receptor 2, which is a well-known biomarker of breast cancer cells, and because of that, a model molecule to be triggered by properly functionalized nanoparticles. HER2 is a tyrosine kinase receptor overexpressed in a relatively huge subset of human mammary carcinomas. It activates signaling cascades which regulates cellular proliferation, adhesion, and migration, and is selectively recognized by the monoclonal antibody trastuzumab, a well-efficient drug usually combined to chemotherapy in the treatment of HER2-positive cancer patients. In the lab, we have designed a HER2-targeted nanoparticle constituted by a magnetite nanocrystal conjugated with a recombinant monodomain of protein A, which has strong affinity for IgG molecules and allowed the oriented conjugation of the anti-HER2 antibody trastuzumab. When injected in mice bearing HER2-positive tumors, such nanoparticles called TMNC were able to detect HER2-positive tumors by MRI, as we can see here by the enhanced negative contrast in correspondence to the xenograft. Injection of fluorescent labeled TMNC in mice bearing HER2-positive tumors revealed that these nanoparticles are able to detect HER2-positive tumors, as we can see here by the fluorescence imaging in both in vivo situations, isolated tumors, as well as tumor homogenates. In particular, a strong signal was present at 5 hours post-injection, as we can see here by the fluorescent signal in the tumor, but unexpectedly, this intratumor signal decreased over time. On the contrary, the magnetic relaxation measured on the same tumor samples revealed a gradual increase of the relaxation rate over time, as an index of the progressive iron accumulation within these tumors. These results at the first glance appear quite contradictory, but then they also suggested a mechanistic hypothesis in which the nanoparticles, once bound to HER2 receptors, were rapidly internalized via a receptor-mediated endocytosis and degraded inside the cells with the disintegration of the fluorescent-labeled organic hormone. This mechanism would explain the drop in fluorescence that we have seen in the previous slide, although the metallic core of the nanoparticle can still be present inside the cell. In order to verify each step of this mechanistic hypothesis, we first verify the expression of the HER2 receptors in tumors extracted from mice upon nanoparticles injection. And by immunohistochemistry, we observed a dramatic decrease of HER2 expression in tumors upon nanoparticles injection, with only a minimal residual amount of HER2-positive cells. This result indicated that the MMC nanoparticle act on HER2 receptor and are able to downregulate its expression on the cell membrane. Then we analyzed isolated tumors by confocal and electron microscopy, and what we observed is that TMMC interacted with tumor cells and were internalized, since it's not that visible, but they are present, they are visible within the cytoplasm of tumor cells at 5 and 48 hours. Electron microscope images confirmed that these nanoparticles are able to interact with the plasma membrane of tumor cells, and they revealed that they then enter an internalization process characterized by membrane invagination, compartmentalization of the nanoparticles within endosomes and then within lysosomes, indicating that the degradation process began within the 48 hours post-injection. These results confirmed what we have previously hypothesized with the mechanistic model by indicating that once reached the target cells, the individual components of the hybrid nanoparticles, that means the fluorescent-labeled organic components and the metallic core, follow differential fate. Then we wanted to try to improve the stability of this nanocomplex in order to achieve a longer-term detection of breast cancer tumors. And in order to do that, we reasoned about how size, shape, and chemistry of the nanoparticle could affect the delivery to breast cancer. And we came up with 3 different hybrid multifunctional nanoparticles composed of a common colloidal iron oxide core, functionalized with 3 different antibody-based ligands. Trastuzumab half chain, obtained by mild reduction of the disulfide bridges between IgG heavy chains, and an anti-HER2 single chain fragment variant containing a cysteine residue within the loop useful for the site-specific immobilization onto the nanoparticle surface. And we then investigated whether and how the structural feature of the immobilized ligand could affect targeting efficiency and biodistribution of the nanoparticles. First of all, the binding capability of these 3 types of nanoparticles was evaluated by flow cytometry, and we obtained an efficient binding capability for all 3 types of nanoparticles. We incubated actually fluorescent-labeled nanoparticles with HER2-positive MCF7 cells, and we analyzed the percentage of cells bound by the nanoparticles. The confocal microscopy confirmed the specific recognition of HER2-positive cells by all 3 types of nanoparticles, and a negligible binding of the nanoparticles to HER2-negative cells. Then we investigated, we compared actually the efficiency of these 3 types of nanoparticles in targeting HER2-positive tumors in vivo, in mice bearing breast cancer, cutaneous breast cancer grown by local injection of HER2-positive MCF7 cells. And we injected fluorescent labeled nanoparticles by the tail vein and monitored their localization for 5 or 48 hours post-injection. The epifluorescence images revealed that at 5 hours post-injection, all 3 types of nanoparticles were able to detect HER2-positive tumors. However, after 48 hours, the fluorescent signal in the tumors treated with trastuzumab conjugated nanoparticles remarkably decreased. Differently, we observed an increase in the fluorescent signal at the level of the tumors treated with half-chain nanoparticles, while no significant difference was observed in case of single-chain fragment variant nanoparticles. The increase in the epifluorescent signal at the level of the tumors treated with the half-chain conjugated nanoparticles was confirmed by measuring the fluorescence intensity of isolated tumors as well as the fluorescence intensity of tumor extracts. In order to try to explain such a different behavior between the 3 types of nanoparticles, we investigated the cellular uptake and internalization trafficking of the nanoparticles by confocal and electron microscopy in tumors isolated at 5 or 48 hours post-intravenous injection. At 5 hours post-injection, nanoparticles conjugated with entire trastuzumab were prevalently present at the cell membrane, while for half-chain and the CFB nanoparticles, the signal spread more inside the cells, suggesting a potential faster endocytosis for these 2 types of nanoparticles when compared to trastuzumab conjugated nanoparticles. Electron microscope images revealed that at 5 hours post-injection, all 3 types of nanoparticles came into contact with the membrane of tumor cells, but then they were internalized with a different rate. Actually, at 48 hours, trastuzumab conjugated nanoparticles were already detected inside electron-dense structures, typically lysosomes, suggesting that they were targeted toward a degradation pathway, while half-chain nanoparticles were still detectable in late endosomes. For the single-chain fragment variant nanoparticles, the degradation rate seemed intermediate since at 48 hours they were present both in endosomes and lysosomes. Then we selected the half-chain nanoparticle for their better in vivo performance and compared their intratumor accumulation with the distribution of identical but unconjugated nanoparticles or nanoparticles conjugated with the half-chain of non-specific IgG. At 5 hours post-injection, a reliable signal was collected for all 3 types of nanoparticles, and in particular for trastuzumab half-chain and IgG. However, at 48 hours post-injection, only in case of half-chain of trastuzumab half-chain nanoparticles, we could detect an increase in the intratumor fluorescence signal, as observed in the in vivo situation, isolated tumors as well again as in tumor homogeneous. Then we concluded that, first of all, multifunctional nanoparticles bioengineered with anti-HER2 molecular ligand induce specific recognition of HER2-positive breast cancer cells and accumulate in breast cancer in vivo. Then, although different types of anti-HER2 molecular ligands revealed comparable efficiency in targeting HER2 in vitro, they show difference in vivo. We have seen that half-chain and single-chain fragment variant conjugated nanoparticles were able to optimize the targeting efficiency in vivo when compared to nanoparticles conjugated with entire trastuzumab, and in particular, only in case of half-chain nanoparticles, we could achieve an efficient detection of HER2-positive tumors at 48 hours post-injection. And this long-term intratumor accumulation of half-chain nanoparticles was correlated with a different internalization and degradation rate of this type of nanoparticles when compared to the other two. Finally, we have demonstrated that the surface functionalization of multifunctional nanoparticles really contributes to the accumulation of nanoparticles in solid breast cancer. Indeed, we have to say that the enhanced permeability and retention effect also in our experiment plays a role and is in part responsible for a short-term intratumor accumulation of the nanoparticle regardless the presence of molecular ligands. But actually the active targeting remarkably impacts on the long-term action of the nanoparticles in vivo. And to conclude, I would like to thank all my colleagues and members of the nanomedicine laboratory directed by Professor Forsi, the Center of Electron Microscopy at the University of Milan, and our principal collaborators from the Nanotechnology Laboratory at the University of Milan Bicocca. And thanks to you all for your attention.
Thank you for a very nice lecture, and it's open for two short questions.
Yes, please. Very nice presentation. How do your results reflect on Herceptin injection itself? I mean, can any of these results apply to modifying Herceptin?
To modify? Sorry.
The original injection of this monoclonal antibody, isn't it Herceptin, the injection?
Okay, we have not performed a parallel comparison with injection of Herceptin alone. But actually we had some other results with a quite different type of nanoparticles where we have performed the trastuzumab Herceptin and Herceptin conjugated onto the surface of nanoparticles, and we saw actually a similar behavior both in terms of accumulation and efficacy. We have some results. I've shown some of them about the the expression of HER2 on the surface of tumor cells, meaning that the trastuzumab conjugated nanoparticles act in the same way as trastuzumab alone, since they bound to the receptor and induce the recycling of the receptor.
Thank you.
So an internalization and a decrease. So they function in the same way.
Thank you.
My question is what is the most important factor of this injection formulations, which increases the internalization or targeting this nanoparticle to this tumor cells? I mean to say the important factors of this injection formulation. Like I know this nanoparticle size or pH. I mean there are many factors also responsible for this. I mean, as per your considerations, what do you think that very important factor for this internalization or uptake of this nanoparticle by the tumor cell?
I don't know if I got exactly your question, but you are speaking about which are the hypotheses for the different internalization rates.
The most important factors of this formulation.
He's asking what is the most important factor for nanoparticles to internalize?
Yeah. Sorry, I got it. Actually, you spoke about size, and this is for sure one of the
Factors, yeah.
of the factors that we have to consider. And actually, trastuzumab conjugated nanoparticles are quite bigger respect to the half chain or single chain fragment variant nanoparticles, and this could in part explain the different internalization rate, since what we know about the literature is that particles which are smaller than 50 nanometers have difficulties to be internalized inside the cells, since they don't trigger an efficient energy for this process. And actually, half chain and single chain fragment variant nanoparticles are around 45 nanometers, while nanoparticles conjugated with the entire trastuzumab are bigger. They are about 75 nanometers. So this could be one of the reasons. Then for sure we have to consider the stability of the nanocomplex, and actually also for this part we have we have seen that half-chain nanoparticles were the most stable ones. So this could be another reason.
Do you
For
consider
sure, these are hypotheses.
Do you consider the pH of the formulation? As these cancer cells have this low pH here.
Okay, for the low pH, actually, these nanoparticles are not that sensitive to the